it's Miss Horseman here, and today we are going to be talking about cellular respiration. Woo! So we have previously talked about photosynthesis and how plant cells create sugars like glucose. Today we're going to be talking about how the mitochondria breaks down those sugars to supply energy to your body. Every cellular activity that occurs requires some type of energy, typically in the form of ATP. So we are going to talk today about how that happens. All right, so this is a very rough drawing of a mitochondria, and I just wanted to show you this overview really quickly. So here we have glucose coming in. We have the first process called glycolysis, okay? The products of that are going to move into the mitochondria, and we're going to have the second step, which is called the Krebs cycle. And then we are going to take all of the energy that's removed in the form of electrons to the third step, which is the electron transport chain. And then we are going to collect lots and lots and lots of energy. So just a brief overview. But first I wanted to talk to you a little bit about the mitochondria. I know we talked about the job of the mitochondria when we talked about cell parts, but we never really got to talk about the structure and the function of it. So if you look here, the structure of mitochondria is kind of, they call it like a kidney bean shape. On the outside, it is a smooth texture, okay, a smooth outer membrane. It's a double membrane organelle, just like almost every other organelle in plant and animal cells. And the inner membrane is very highly convoluted, which means it's very folded, okay? The cool thing about that is the more folds that there are, the more space there is for all of these cellular processes that are going on. So it increases the surface area, okay? All right, and then we've got our inner membrane fluid, which in this case is called the matrix. Okay, so glycolysis and fermentation occur just outside the mitochondria in the cytoplasm of the cell. And then the second two steps, Krebs cycle and the electron transport chain, are going to occur in here. The Krebs cycle actually occurs in that fluid area, the matrix, and the electron transport chain occurs in that it's called the cristae, which is basically the inner membrane. All right, so let's get down and dirty. Cellular respiration is a process where cells transfer stored energy from what the organism has eaten. So when I ate pizza tonight for dinner, pizza has crust, which is made out of wheat, so it's a carbohydrate. Tomato sauce, which is made out of tomatoes, a plant, also a carbohydrate. I had mushrooms on it, so that was a fungus, which is equivalent to a carbohydrate. And then I had chicken on it, which is a protein. So all those carbohydrates that I ate for dinner are going to be digested over time, and then it's going to be broken down into the raw components, the very smallest components, which for glucose, or for carbohydrates, is glucose, okay? So cellular respiration is going to take that glucose and it's going to break it down and give my body ATP, which is the energy source that it can use. So remember we talked about glucose is too big for a cell to be able to use it for energy. That would be like me going to a soda machine and trying to get a soda out of that machine with a $100 bill. That $100 bill is too big. So our body has to make change for that glucose molecule, okay? It has to break it down into smaller energy components. Like I would have to go and get change for that $100 bill. So I would break it down into dollars or even quarters, and then I would put in what I needed to get the soda out, okay? Our body can't use that glucose molecule. It's too big, so it has to break it down first to get the energy components out of it that the cell can use, okay? There's actually two types of cell respiration. The first one is aerobic. And when I hear the word aerobic, it automatically makes me think of like Jane Fonda doing step aerobics or whatever. Aerobic means with oxygen. When you're doing workout aerobics, you are breathing heavily, okay? So with oxygen. Anaerobic, anytime you put an as a prefix in front of it, it means not. So anaerobic means not oxygen or without oxygen. So that's the process that happens when there's no oxygen available. And we're going to talk about how both of those occur. All right, so the very first one we're going to focus on is aerobic respiration, aerobic with oxygen. If you look right up here, I have the equation written out for you. in words and in the molecules. So we have 6 oxygen plus glucose yields or gives you 6 carbon dioxide, 6 water, and a ton of energy. Okay, this is the biggest part of that whole process that we're focusing on. So what are the reactants of cellular respiration? Don't forget that the reactants are the ingredients. They're what goes in at the beginning, and then the products are the outcome that comes out at the end. So here are the reactants, here are the products. I hope that looks a little bit familiar to you, especially if you paid attention to the photosynthesis video. This should look exactly like the photosynthesis equation, but flipped backwards, okay? So hopefully that'll make it a little bit easier for you to remember. It's the photosynthesis equation, but it's flipped upside down, okay? So the very first step of aerobic respiration is glycolysis. And glycolysis literally means to split glucose. Glucose, lysis means to split. So that glucose is going to literally be cut in half, okay? In order to get this process started, your body has to use 2 ATP. So I kind of think of it like those little penny cars that I used to play with when I was a kid, or the little cars that you had to like rev up a little bit in order for them to go. Your body has to give a little bit of energy. It has to rev up this process in order for the process to take off and go, okay? So we have to give a little bit of ATP molecules in order to get some ATP, more ATP molecules out of it at the end. Another way you could think of it is going back to the soda machine. If I put 2 quarters into the soda machine, I get my soda back and I get change back. So I put in 2 quarters, I get my soda, and I get like $1 back in change. Awesome! That would be the best soda machine ever. So in this case, in the case of glycolysis, we're going to use 2 ATP molecules, but we get 6 out of it. So we say it makes 4 net, or 4 total ATP, okay? It also makes 4 electrons. It strips those electrons off of the glucose, and it makes 2 full electron carriers, NADP, okay? It's very similar to the electron carrier that we saw in photosynthesis. This one's called NADP. It's specific to the cellular respiration process. Okay, this can occur in the absence of oxygen. So it doesn't need oxygen for this specific step to occur, all right? So we split glucose in half, and out of it we get, let's take a look at this. Here's our glucose molecule right here. It's got 6 carbons, okay? It's going to split it in half, and here's the use of our ATP molecules, so we're going to use a little bit of that energy, and we literally broke that glucose molecule in half, okay? Right here we've got some of our electrons being stripped off, so we made 1 NADH molecule, and since we've got 2 of these new molecules, we're going to have NADH down here, and we're going to have NADH up here, okay? We're going to continue moving through that process, and look here. We make 2 ATP here, and we make 2 more ATP here. And at the end of the whole process, we have made a little bit of energy, but we still have 2 molecules left. These are called pyruvic acid, and that's going to be important in the next step, and I'll show you in just a second. Okay, so the first step is glycolysis. The second step is the Krebs cycle. At the end of glycolysis, there's still a ton of energy that's still left in those pyruvic acid or pyruvate, whichever one you want to call it. There's still a ton of energy left in those molecules. We, by far, our bodies have not taken all of the energy that was available out of there. So if oxygen is present, that pyruvic acid is going to move on into the next cycle, which is the Krebs cycle. It goes into the mitochondria, and it's going to be broken down further in a series of steps, okay? I'm not very specific in that you know all of the steps of the Krebs cycle. What I really want you to know is that as that cycle moves around, carbon dioxide molecules are going to be pulled off, okay? High energy electrons are going to be stripped off as those carbon dioxide molecules are pulled off. Some of the electrons are pulled off too, and those are going to go to some new electron carriers. All right, so here is a process, a picture of the whole process. We've got our pyruvate that we got from glycolysis, okay, so that's coming in. We have our pyruvate. Look here, carbon dioxide is being stripped off right there. So are some electrons. So we've got an electron carrier, NADH, right here. Now we have a 2-carbon molecule, and that 2-carbon molecule is going to move into this cycle. It really is a cycle. It goes around and around. So some of these molecules are going to be recycled over and over again. All right. We have another carbon dioxide being stripped off. As that stripped off, we have more electrons being pulled off and passed on to these electron carriers, NADH. Look here, we've got some ATP being made, just a little bit of ATP being made there. And then as this process continues to go so it can restart again, we have electrons being pulled off and it's given to this electron carrier. It's called FADH2. All you need to know is that it's an electron carrier just like NADH, okay? So we have broken the rest of that glucose molecule down so that all that's left is something that's going to be recycled and the carbon dioxide that our bodies get rid of in the process. But we have made a little bit of ATP and we've made a ton of these electron carriers, NADH and FADH2. And because that glucose molecule is broken in half, this whole process occurs 2 times for every 1 glucose molecule. All right, so far what have we made? For 1 glucose molecule, we've made 6 ATP, 4 from the glycolysis and 2 from the Krebs cycle. We've made 8 NADH, and we've made 2 FADH2. Now these molecules are going, the NADH and FADH2 are going to be used in the next process that we're going to talk about. So we've made a little bit of that ATP energy, but we have made a lot of these electron carriers, and that's going to be really important in the electron transport chain. Okay, so this is actually very similar to the electron transport chain that we saw in the thylakoid membrane. So this is the mitochondrial membrane on the inside, okay, that really folded membrane that was on the inside of the mitochondria. Here we have all of these proteins, okay, that are stuck in this membrane. And then this one should look familiar to you. This is ATP synthase protein. Okay, so the electron transport chain uses those energy carriers, NADH and FADH2, and they're going to drop off. You can see here it's dropping off the electrons, so the electrons move here. The NAD plus now, because it's been stripped of its electrons and its hydrogen, the NAD plus is going to go back to the Krebs cycle so it can pick up more electrons, okay? Now look here, we've got all of these hydrogens that have been stripped off the NADH molecule. But we need to make sure that the outside of this is a low hydrogen concentration. So as these electrons are being moved down that protein, electron transport chain of proteins, it's going to actually use that energy from the electrons to pump the hydrogens across the membrane. So now we've got this really high concentration gradient of hydrogen molecules, hydrogen ions. That is super important because we know facilitated transport moves molecules from high to low concentration through a protein in the membrane, okay? This ATP synthase protein relies on that hydrogen concentration gradient because as the hydrogens move through here, it's just like a turnstile like we saw in the last time. It's going to spin that ATP synthase protein around just enough to take an ADP molecule and a phosphate, and it slams them together and it makes ATP at the end of it, okay? So this happens for every one of those electron carriers that we made in the Krebs cycle and in glycolysis process, okay? The hydrogen ions do not join to the ADP molecule. That is a very big misconception I want to clear up right now. Those hydrogen ions do not add to the ADP and phosphate molecule to create ATP. They're just going through that ATP synthase to cause it to spin. So once they come out here, they're either going to join up with the oxygen that we're going to talk about in just a second, or they're going to be pumped back across the membrane, okay? Now, oxygen. Those electrons fell down the electron transport chain, okay? And as they did, they released some of their energy, but they have to go somewhere. I told you before that electrons can't just go floating around in our cells. It would create total chaos and it would destroy them actually, okay? So something has to pick up those electrons. They're not needed anymore. So our bodies use oxygen as the final electron acceptor. So there's oxygen hanging out here just waiting to pick up these electrons. After it picks up electrons, it's going to become negative molecule. So it's going to be O2 minus, okay? We know negative is attracted to positive. So we have these hydrogen ions that are hanging out here just waiting to do something. So we've got now a negative oxygen molecule and some positive hydrogen ions. They're going to join together and they are going to create water. Now, this process can continue until it's out of oxygen. If that oxygen is gone, then the electrons back up on the electron transport chain because they have nowhere else to go. If these back up on the electron transport chain, NADH and FADH2 cannot drop off their electrons anymore. So that means the Krebs cycle gets backed up and the whole process stops. Okay, so that's why it has to have oxygen in order for this process to occur. Aerobic respiration with oxygen. All right, so this kind of tells you exactly the process that I walked you through. The energy, the electrons from NADH and FADH2 is passed to the carrier molecules in the membrane of the mitochondria. That's the electron transport chain. The oxygen that's brought into the cell is combined with those electrons that have fallen down the chain and a hydrogen ion to make water. Every time 2 electrons move down the electron transport chain, those hydrogens are pumped across the membrane in a protein pump. And because now we have a high concentration gradient of the hydrogen ions inside the membrane, it can be used to move through the ATP synthase. This causes the ADP to bond to a phosphate, and it makes ATP. All the electrons from one glucose molecule make enough energy to create 34 ATP. So for every itty-bitty glucose molecule that's broken down in the presence of oxygen, it makes enough to make 34 ATP, and I'm going to show you exactly how that happens, okay? So I'm going to take you through this really quick again. Glycolysis is the first step. That glucose molecule is broken in half. We use 2 ATP, but we gain 4 out of it, okay? So that's where it's showing here 2 ATP because it's taking the total, okay? So 4 minus 2 gives you 2. All right, we also have NADH molecules that are coming off of this. Here's a little mini step that's going into the Krebs cycle. We have more NADH being pulled off. So this pyruvic acid moves into the mitochondria. We have more NADH pulling electrons. The Krebs cycle moves around. We have more NADH pulling off electrons. We have FADH2 pulling off electrons. Every time one of those carbon dioxides are stripped off of those pyruvic acid molecules that's moving around in this Krebs cycle, we get more electrons being stripped off. We also get a little bit more ATP that's being made. Now, all of these electron carriers are going to the electron transport chain, and this is where we get most of our ATP being made, okay, through that whole process, the hydrogen concentration gradient, those hydrogens moving through the ATP synthase, okay, and it makes about 34 ATP. So the maximum amount of ATP that we can make per glucose molecule is 38. That's a ton. That's a ton. But remember, your body, your cells use these ATP very quickly. Your cells actually only have enough ATP to last for a few seconds. So this process is constantly working in your bodies to make sure you have enough energy to do what you need to do. So go ahead and pause the video really quickly. I want you to look at these questions and make sure that you have an idea on how to answer these. Okay, if you don't, you need to go back and watch this part of the video again. If you have questions, we will be sure to go over them in class, so write them down and make sure that you know what you want to ask. So that was aerobic respiration. Now I want to talk really quickly about anaerobic respiration. If no oxygen is present, the cell goes through glycolysis, but then remember I told you that the whole electron transport chain and Krebs cycle gets backed up, so it can't do the rest of that process anymore. Okay, so instead it goes into fermentation. Fermentation releases energy from food molecules in the absence of oxygen. Okay, and there's 2 different kinds of fermentation. The first one is alcoholic fermentation, and it's produced by yeast and other microorganisms. So what happens is, in an environment where there's not any oxygen, the glucose is broken down, and it produces alcohol and carbon dioxide. Okay, it provides a very limited amount of energy for those microbes like yeast, but they're really small, they really don't need a lot, and they don't live a super long time either. So let me show you what that looks like. This should look familiar. We looked at this process when we were looking at glycolysis. Here's our glucose molecule. We're going to use a little bit of ATP. We're going to split it in half, and now we get a little bit of ATP out of it. We also get some of those electrons being stripped off. So here's our pyruvic acid. Okay, but now there's no oxygen available. So instead of going into the Krebs cycle, it's going to go into the second part of fermentation. Okay, so we've got carbon dioxide being pulled off. We have some electrons being stripped. Those electrons are, we have electrons being added, electrons being added so that NADH can go back to pull off more electrons. So this NADH is going to get recycled through this little process. So in the end of it, we have a little bit of ATP, we have carbon dioxide that's released, and we have ethyl alcohol. This is how beer and wine and other spirits are made, okay? So when you see on the commercials, because I know you don't have firsthand experience for this, when you see in the commercials the beer being poured into the big glasses, you see all of these bubbles coming up, right? And it makes like a head at the top. That is the carbon dioxide that was created from the yeast that were going through this whole process. All right, the other one is lactic acid fermentation, and this directly affects you because your muscles do this when you do any kind of exercise, okay? So glucose is turned into lactic acid and a little bit of ATP. This occurs during exercising because your muscles can quickly run out of oxygen, but it still has to produce some kind of energy, okay? So it produces a little bit of ATP by lactic acid fermentation. Your muscles can't sustain this activity for very long periods of time. What actually happens is this lactic acid builds up in your muscles and it causes a very intense burning sensation. So for all of my baseball and football and soccer and softball, basically anybody who plays sports out there, after you're done with your sport, a lot of times your coaches should have you do some kind of cool down activity, and it might be just jogging around the gym or jogging around the track. What that's doing is it's actually causing your heart to pump and breathe, and it's causing that lactic acid to flush out of your muscles. If you don't do that, you're going to be sore for sure in the morning. So make sure that you cool down after you're done with any type of exercise. So here, really quickly, is that process. The glucose molecule is breaking down and making a little bit of ATP. This NADH molecule is stripping a few electrons off of it. Now we have our pyruvic acid, but we have to donate those electrons back so that we have more NADH available to go and do this process again. So by doing that, it makes 2 lactic acid molecules, and that's what makes your muscles burn when you're doing any type of intense exercise. So the last thing that I want to talk to you about is I really want to refer back to this cycle. So we talked about photosynthesis, and we know that photosynthesis requires carbon dioxide and water in the presence of sunlight in order for this chloroplast in a plant cell to make glucose and oxygen. Now that glucose and oxygen is what's needed for mitochondria to do cellular respiration. And out of that we get ATP, energy for our cells. We also get carbon dioxide and water. So I want you to see that this is really a cycle. The products of photosynthesis become the reactants of cellular respiration. And the products of cellular respiration become the reactants of photosynthesis. And on and on and on. You can't have one without the other. So that's cellular respiration in a nutshell. I know this is quite a long video, but I hope that you saw how important it is for your body to get glucose molecules in order to give your body, your cells, the energy that it needs, because they won't work if they don't have the ATP. Make sure you have any questions, you write it down, and I'll talk to you soon.